Okay, this is our first lecture on chapter 22. That's the chapter on prokaryotes. And so we're going to talk about the different types of prokaryotes and their characteristics and their diversity. So should be, these are some of the simplest organisms. They're all single cell, and so you really can't see them individually, but we know they're there and we know they're abundant. And so I have two pictures here to kind of show you. This one picture is a woman and she's standing by a dried up pond, and or excuse me, a hot spring, and the organisms that are leaving these colored marks here. These are a film of microorganisms. It's not a mineral deposit that happened naturally. It's microorganisms that growing there. And even closer to home, here's a dental shot from someone who hasn't been brushing or flossing, and you can see on their teeth there's this film. Okay, that's what the dental hygienist scrapes off. That's plaque, and that's made by individual bacteria secreting a sticky substance so they can stay attached to the surface of your teeth. And so these, we can't see individual organisms, but we can see them in their millions, forming films and changing the color of water and various things like that. And they're abundant. They can have big effects in the environment, and they're important. Um, Bacteria, most of them are very, very small, in the neighborhood of maybe 1 to 5 microns or micrometers in length. Um, the smallest are about zero point zero 0.02 microns, so something like, you know, um, 100, 200 nanometers. So really, really small. The largest are almost a millimeter in length, but they're mostly really, they have storage vacuoles in there. Um, Bacteria, how many different kinds of bacteria are there? That's really hard to say. At first, the only way we could find bacteria was to grow them in culture on agar plates or in broths like you see down here. Um, but later on, we've been able to figure out ways to get indications that there are lots of species there that we can't grow. We do this with something called polymerase chain reaction, where we use a probe to fish out ribosomal genes out of the soil. And so we look for 70S ribosomal genes, and then we sequence them and see how many different types there are. These all cells have these genes, and so we're looking for organisms that have their own unique varieties of them. And we find that just a gram of soil may have 10,000 different types of bacteria in it, so they can be very, very abundant. It's not unusual for people in their intestine to have 1,000, 2,000, 3,000 different types of bacteria grown in their intestine. Lots of these, all we know about them is the kind of ribosome they have, because we can't grow them. And then we haven't sampled every environment. Bacteria are really small. Most of them we can't culture because they need special nutrients, or they may be part of complex food chains. But we know there's lots of them, that they're abundant, and there are many different types. Okay, this is just a kind of a review slide for cell theory. Okay, all organisms are made of cells. Life processes occur within cells. Prokaryotic cells are the smallest types of cells. And we think these cells appeared first. They're very simple, small, simple, with no internal compartments, no nucleus, no internal organelles that have membranes around them. It's just a single compartment with cytoplasm, a DNA molecule, and some ribosomes in there, and then lots of enzymes. Unfortunately, they all look alike to us mostly, but metabolically they're very diverse. Okay, and so those are the prokaryotes. When we do a phylogeny, okay, we find one domain of prokaryotes is more closely related to us than the others. And then the second domain of prokaryotes is a little more distantly related to us. Okay, I like this, this kind of shows a clade organization, whereas your book uses this one. So a universal ancestor gave rise to archaea and eukarya and on one branch, and then the other branch was the domain bacteria. Okay, um, bacteria are old. Okay, we have fossilized structures that go back over 2 billion years called stromatolites. They look identical to formations that occur in very salty waters off the coast of Australia. And these are bacterial mats that just grow in this salty water. And if you section them, they look just like some fossils that we find in very, very old rock. So that's a stromatolite. You should know what a stromatolite is. It's good evidence of the early cells, and those cells are prokaryotic. So your book gives us a little kind of insight into prokaryotic structure here. Um, Prokaryotes, in general, the cell is all prokaryotes have a rigid cell wall surrounding the cell. Just inside of that is the cell membrane, and then a single compartment of cytoplasm that contains a single DNA chromosome, a loop. It contains some ribosomes that are always there, and then lots of enzymes. Prokaryotes may or may not have a flagella for locomotion, and they can have more than one. They may or may not have these extensions called pili for attachment, and they may or may not have a capsule, which is a kind of a slime layer that helps them stick to surfaces and protects them. And so, like with the plaque on people's teeth, that's that capsule. All right, prokaryotic cells, they're not that interesting to look at because the cell wall determines the shape, and there's only several basic shapes, and they're shown here. These round shapes are called cocci. That's for plural. An individual is a coccus, and it's a sphere-like structure, and these can be a variety of different sizes. That's very common. Another extremely common shape is the bacillus, which basically looks like a hot dog. And again, bacilli is plural. The singular is bacillus. And then the third shape is a spiral shape, and this can go by a couple different names: spirilla, spirochete. They're like that, and some organisms have this. And these shapes are determined by the cell wall of the bacteria. What distinguishes prokaryotes is that they may have different types of cell walls. Okay, the material that forms the cell wall in bacteria is called peptidoglycan. In archaea, the cell wall material is similar but different. It's called pseudomurin. And this is really, really good. It can separate all different organisms. We used to use a staining method called gram stain, and actually we still use it to give us what type of peptidoglycan is present and how much. Also, the two different types of prokaryotes have different types of ribosomal RNA. This one is the ribosomal RNA is slightly smaller. It's referred to as 70S. Archaea have a ribosome similar to ours. It's called an 80S ribosome. Okay, and then the gram-negative and gram-positive refers to how much peptidoglycan is there and the presence of other lipid layers. The cell membranes also differ from archaea and eukaryotic or bacteria. There's a different type of linkage between the one group has an ester linkage, which I believe is the bacteria and eukarya, and the other group has an ether linkage. So there's these differences. That's kind of the basic overview. We're going to go into more detail in the different groups next time. I thought I'd just show you a couple quick pictures. These are electron microscope pictures, so they give very fine detail. It's a group of mixed bacteria, so you see they've been given different colors. There's a couple different bacilli there, one with lots of pili on its surface. There's another one that's kind of has a flagella here shown in pink. There are orange cocci in this guy. Another picture here, again, electron microscope showing different types of bacilli. They're given different colors. Here's a coccus right here, and then several different types of bacilli. Um, and again, we're getting a size bar. These are about 2 microns, so much smaller than eukaryotic cells. Because they're small, prokaryotes can reproduce quickly. They can reproduce extremely quickly, like 10 minutes in some of them under good conditions. There's a nice picture of a bacillus bacteria. Here's Staphylococcus aureus. Here's with a light microscope here, and they're just dots. And these are these little purple dots here. This shows you with an electron microscope so you can get more resolution. This causes infections in people. E. coli also sometimes causes infections, but most of us have a strain that's non-pathogenic in us, and so we all have this. It's just whether we have a pathogenic or non-pathogenic. Again, this is a bacillus, and these are flagellae that are tangled together. Here's a nice picture of a leptospira. So it's a spiral-shaped organism that you see right there. There's a spirochete, another guy right there. You can't really tell the difference between bacteria and archaea. Archaea look the same. Here's a coccus that's archaea. They are, again, these guys are, they're single-celled. All of the archaea are anaerobes. They can't tolerate oxygen, but they can tolerate heat and acidic pHs, and they can do weird things. Here's another coccus, a methanogen, and here's a methanococcus. Okay, so next time we'll go over the different groups of bacteria and archaea in more detail. There is a Moodle quiz that's open, and so you can start on that.